Today, we have the distinct pleasure of having two very impressive scientists join us from the DRI in Miami. Dr. Chris Fraker and Dr. Allison Bayer are joining us, and I'm just going to give a short little bio about the both of them. Dr. Fraker, I will start with you. He's focused on optimizing islet stem cell encapsulation to restore normal glucose levels without the need for immunosuppression, sort of like what everyone is hoping to achieve in this field, really. And additionally, everyone's working very hard. By that, I mean that people are working really hard towards that end. And additionally, he continues to work on improving oxygenation to encapsulated cells and applying these technologies to the growth and maintenance of mesenchymal stem cells, or MSCs, islets, immune cells. The nanoemulsion technologies developed in Dr. Fraker's labs are also licensed for development as injectable anesthetics, repurposing inhalation anesthetics into the nanoemulsions, which is very cool. And more recently, he's begun work on the role of the innate immune system, natural killer cells, or NK cells, and T1D, and the importance of these cells in the viral hypothesis or hypotheses surrounding diabetes development. He's also examining the importance of innate immune function in autoimmune pathologies and the existence of precursor cells in MSCs. And he's involved in projects in examining a potential metabolic factor in both T1 and T2 diabetes, which is folic acid. As reported in the Journal of Immunology, Dr. Allison Bayer's team established an important function for IL-7R, or otherwise known as the IL-7 receptor, for the development of natural Tregs, or T regulatory cells. This will be an important line of future investigation, as polymorphism in the IL-7RA gene that likely leads to reduced IL-7R signaling is an autoimmune susceptibility gene for both multiple sclerosis and type 1 diabetes. And Dr. Bayer's research focuses on understanding the basic immunobiology of Tregs and applying that knowledge for future clinical translation application. She hopes her work is going to lead to the design of novel therapies for non-toxic approaches to tolerance induction, with the ultimate goal of achieving both self-tolerance and transplantation tolerance for the treatment of T1D patients. Welcome, both Chris and Allison. Thank you very much for taking the time out of your busy schedules to speak with us.
Thanks very much for having me.
Monica.
Yeah. Okay, do you want to dive in here? The title of this presentation, The Role of NK Cells in the Context of Type 1 Diabetes, is it the beginning of the end? And as I said to you both earlier, I really hope so. That would be great. But yeah, let's talk about the work.
Okay. I'm going to start off just with a little personal story and sort of parlay into Allison's real research work. I am by no means an immunologist. I'm a bioengineer by training, but I'm also a T1D patient for 36 years. And in 2009, I happened to get a flu vaccine, which sent my immune system spiraling, for lack of a better word. I reactivated about 7 viruses. Wow. And was basically stumbling around. I got Guillain-Barré syndrome and was basically stumbling around looking for answers to what was going on. I had horrible chronic fatigue, which is now being called long COVID, even though it's existed for over 50 years. And I finally found a physician here at UM who treated patients in this chronic fatigue and immune dysfunction. She's one of the world's leading experts. Her name is Nancy Klimas. And my doctor, Irma Ray, who works with her. And they basically told me that a lot of these illnesses that are caused by viruses are problems in the immune system of patients, basically the early immune system or the innate immune system, and primarily NK cells, the natural killer cells, whose job it is to deal with viruses when they first come in, along with a couple of other cell types. They're the ones that are responsible for getting the whole immune cascade started. And she said that all of her patients typically, and interestingly, when I started to research and looked at autoimmunity and other conditions, it seemed very similar. All of them tend to have defective NK cells. They don't function properly. They function in a way that's actually immunosuppressive, for lack of a better term. They shut down other immune responses. And it seems that these viruses, which I thought, you get a viral infection and you clear it. No, that's not the case. They live in patients for years and years, their entire lifespan. They can be passed on to their children, right? So you have these viruses living in you. These are viruses of the herpes family, like Epstein-Barr, cytomegalovirus, HHV6. These are viruses of the Coxsackie family, B4, B6. These are viruses of many different families. And many of these viruses you've heard in the context of autoimmunity, because there have been reports for over 20, 30 years of people seeing these viruses tied somehow to people with autoimmune conditions. And that sort of led me to find Allison and ask her, we had been colleagues, and I'm like, look, I really want to figure this stuff out. I really think this is something to do with diabetes and with other autoimmunity, and what can we do to start studying this? And so, of course, being the great collaborator and generous colleague that she is, she decided to look at my crazy ideas and try to figure out some of this stuff. So that just sort of moves forward into some of what I was talking about. So here are some recent papers. Most importantly, this one at the top left, which was a recent paper basically describing in patients that they did a study with military blood samples, and they found that people that at some point had EBV and had reactivated for EBV were 20-fold more likely to develop MS following long-term study. So this was really considered, and this was published in Science. I think it's the left one, I can't see. But anyways, this was one of the definitive papers that is establishing that, these viruses are tied to autoimmunity. And more this is growing as techniques are getting better to detect the viruses, and samples that could only be measured in one lab now with better techniques, they can be confirmed in other labs. And this is part of the, the great program of UF and UM and other centers, the NPOD, where they're taking pancreatic donors that had type 1 diabetes and controls as well, and they're comparing, and they're seeing these the viral presence in different pancreatic samples, which is really intriguing, and they're finally able to confirm it. So NK cells, they weren't really, most of the research in immune cells in diabetes, in fact, like 95 to 99% of it has been done on T cells, and particularly CD8 positive cytotoxic lymphocytes, or T cells, which are the very downstream end effectors of the response. And one of the interesting roles of NK cells in the body, if you have, for example, an influenza infection, is to control the response of the CD8s, to make sure that it doesn't get out of control, right? So there are papers published showing that when people that have conditions like diabetes and their NK cells are not very strong, they get the flu, those CD8s go crazy, and they cause too much inflammation, too much tissue destruction, and you get a very severe reaction to the flu. Well, thinking in the context of type 1 diabetes, our hypothesis was, yeah, that makes sense, because those are the cells that destroy the pancreas. And if they're not being controlled by the NK cells, and there's some missing communication from the early immune system, that basically the prehistoric immune system, right, that further developed when viruses were able to evade it, it makes sense that they would destroy the pancreas, and the NK cells, if they're not working right, you're going to get some destruction. So that was,
yeah, I mean, so Chris, it
yeah.
looks like from these papers that this hypothesis has been around for a while, kind of
For
floating
sure.
around. Yeah, right?
Yeah, but undefined, right? Like, so the technology
Yes.
I mean, for sure, viral hypothesis has been there forever. We just kind of tried to tie some pieces together and see if it made
Right.
sense. And we threw it out there because we want people to pick it up.
Yeah,
And
no, I think it's so fascinating. Like you can see there's a buzz around the NK cells and what is their role in, the initiation of T1D. And I think it's really interesting how you kind of started to put these together.
And only
Well,
because I got that shot, right? Like, I mean,
yeah.
the thing that's exciting to me too is COVID is pushing some of that out there too, right? Because we've had this surge of diabetes and of this long COVID. So now, like, in some states, 41% of the population is claiming they have long COVID, right? And this is something that was basically ignored for years. It was a very, the way it was described was very demeaning. It was called housewives disease or hysteria, right? Because the people that primarily got it were women in large disparity, the same with autoimmunity. And the reason is if you think about it, every month from the age of menstruation to menopause, they basically are immunosuppressed, waiting for implantation. And that allows viruses to reactivate and to do all these things. So that was part of our hypothesis as well. This is why it so disproportionately affects women, we believe. And in the case of diabetes, where it's equal, there is a specific reason for that as well that we know now. There's a marker on the beta cells that basically gives immunoprivilege to that cell for viral infection.
It's
I just
fascinating.
also want to add, I just want to add that we're always talking about the hyperexpression of MHC class 1. And Pod has confirmed this, it's been discussed, but we talk a lot about the CD8 cells, but the other immune cell that really uses and hones in on MHC class 1 is NK cells. So to kind of ignore this very innate cell population that is also being led to figure out, do I react, do I not react, is tied to MHC class 1 expression. So I think as a field, we definitely need to think about the role of NK cells and how it actually is contributing to the disease in the patients themselves, but also, how do we use these populations to really understand the pathology? So that's kind of where like Chris and I, started with this project.
Yeah, that makes
Yeah.
sense.
Yeah, I mean, and Allison was, I mean, like it was great because even my physician was excited about it because she sees this potential too. So it gave us two sort of hypotheses in the sense of there's one or two things we think are happening to NKs. They're either, they're just constantly exhausted, which is she can describe in better detail because there's a constant source of inflammation. There's a constant viral presence that they're detecting through cycles of reactivation, then going back into a latent state. And that just wears them out over time. And that's what my doctor always says when she looks at my NK cell profile, my viral profile, she'll be like, yeah, you're suffering from really exhausted NKs. Their signaling molecules are all disproportionate and out of whack. So these things called cytokines that they use to communicate with other cells are completely off, right? And they're just worn out. And then they're also being used by the viruses. They're going through these cycles of destruction that we ended up seeing in the patients where they become more suppressive and the viruses sort of hijack that mechanism to prevent being detected, right? And so then
this whole model fits, very nicely with the whole like one hit and then remission, one hit presentation of an autoantibody remission, so-called remission, and second hit, another autoantibody, a potential remission, and then finally diagnosis. But, I wondered, have there been any larger scale trials that are mapping the NK cells of patients as they are in the prodrome?
that's the overlooked cell, right? I mean, that's what I think now people are starting to pay attention to stuff. I mean, even the samples that I had it was a very small number. I mean, we were able to show in that small population with age and sex matching that there was some significant difference to controls. But the amount you would need to do and should have been done, right? They should have looked at, but I mean, a lot of it is the technology too, right? They didn't have the staining for the antibody or the antigens on NK cells that they do now, right? So now they so
or the
many
seq scope
subpopulations.
or anything like that.
Yeah, I mean, there's so many subpopulations defined now as well that have newly discovered roles. I mean, in the last 20 years, the advancements made in that are amazing. And that's why I think it's going to get better. And even the technology around looking at those cells is getting better too, like the flow cytometry techniques, the machines, everything. I think there's going to be a lot of discovery in the next 10 to 15 years.
How difficult how
Yeah,
difficult
go
is
ahead.
it to isolate sort of NK cells from a patient's blood? Are they very easy to get a bunch of them to be able to study, or are they very rare?
Yeah, it's fairly easy to get the cells. The hard part is keeping them, like I would run my samples fresh. So I had the benefit of the clinic downstairs where we would get patient samples and then I would bleed every person here that didn't have diabetes that were my colleagues and such as my controls and other controls. And then you could compare, and then you could even buy whole blood from a blood bank, things like that. And if it's fresh, it's pretty easy. They do degrade fairly rapidly, at least in my experience and what my doctor had said. So I don't know if there's literature out there now that says they found better ways to keep them viable for longer. But so now we're getting into the area. I'll let Allison talk about some of her work where she was looking at the NK cells in mice, both in the controls and in the autoimmune population. Some really interesting stuff. So
Sounds
go ahead.
great.
Thanks, Chris. So yeah, I mean, typically when we think of human natural killer cells, we think of two subsets. So sometimes they come in different flavors. People kind of refer to them with different names. You have the CD56 brights, which some people will call NK regulatory cells because they make copious amounts of cytokines. and they can kill, but they're not as proficient as the NK effector cells. And we kind of know some of the expression patterns in these 56 brights. So we have, they express the trimeric IL-2 receptor, they express c-KIT, which is supposed to be, you think of more in a more immature phenotype. They have L-selectin, CCR7, and of course, then they have the related, inhibitory and activating receptors on the surface. And then, but most people, when you talk about natural killer cells, they think about the NK effector population, which is the CD56 DIMs. These are the killers. These are the ones that are doing the destruction. They have lots of granules in their cytoplasm and they're full of killing molecules. They're very good with natural cytotoxicity as well as other forms of killing. And they do make cytokines, but not to the same extent that these NK regs or 56 brights. Yeah, Chris is gonna have to use the pointer because my point got the slides going. So typically when we think about this, most people think about NK effector cells, but the 56 bright cells are also very important. These are what regulate the adaptive response through their cytokine secretion. And we also know that these NK reg cells, though they're not very good killers, when they become killers, they're very good at killing effector CD8 cells. There's more data coming out about that. So it's definitely where we need to be thinking about subsets. Chris, you can switch the slide.
Okay.
Yeah, so what Chris and I first started doing is, as you mentioned in the beginning, I'm really a T regulatory cell lab. So when we kind of delved into how are we going to look at the phenotype of these NK subsets, and we want to look at control subjects, and we had relatively easy access to blood samples from long-standing type 1 patients. So we developed this strategy using flow cytometry where we could identify the NK cells and very clearly identify these 56 bright CD16 negative compared to the CD56 dim CD16. And you can see here on the flow cytometry plot, we kind of back colored those. So the NK regulatory or 56 bright are red, where the NK56, the CD56 dim NK effectors are blue. And we kind of looked at some typical markers. If you look in panel B, we looked at CD27 and CD11B, and we immediately noticed that the long-standing patients have a propensity to have a higher proportion of these more immature like NK cells. And you can see, we see both populations that are blue and red dots indicating they're both 56 brights and CD56 dim within that gate. And when we look at panel D and we look at 56 expression with granzyme B, you can see that the 56 dim, the blue cells, are the ones that are mostly expressing the granzyme B or the killing molecule. Though that the long-standing patients also have it, you can see they seem to be less robust in the staining. And the 56 brights really don't have granzyme B expression, at least when you take them directly from a fresh blood sample without stimulating them ex vivo. But what we wanted to look at is kind of this idea of immature NK cells versus mature NK cells. So we wanted to use c-KIT. It's kind of a distinguishing marker, which most people don't really delve into the differences using this marker, but we thought it could potentially be an important marker when we think about the activation status and the roles that these different subsets can be playing. So you can see in panel C that really it's the 56 bright cells that are expressing the c-KIT. And again, we see this propensity of long-standing type 1 patients to have a higher proportion of this subset when we're gating on the total NK population within the peripheral blood sample. And again, we see a similar pattern when we look at c-KIT versus granzyme B to what we saw with CD56. So c-KIT is a good marker for kind of looking more at these immature NK cells. If you go to the next slide, Chris. There you go. So we actually, as Chris mentioned, we looked at a handful of patients just to see, could we see differences in the subsets of these NK cells in the peripheral blood of either control, long-standing type 1 patients. And we had a small number of at-risk patients, so autoantibody positive patients. And if you look at the left panel, you can see even between the control and the long-standing, when we looked at the NK effector, so the CD56 dim, CD16 positive cells within the total PBMC, we have a propensity to have less of these cells, which again means we have more regulatory cells, or 56 brights. And when we subdivided that between age and sex, that's what you see on the right side. You can see that the trend holds true for the long-standing patients, that you have this overall decrease in this relatively small number. Again, we would love for clinical trials to really delve into these subsets in more detail so we could kind of get a better understanding how therapies might be affecting this population. And although we had a small number of at-risk patients, you can even see at the at-risk patients have a propensity to have a lower level of NK effector population in the peripheral blood lymphocytes. So we found that interesting. Again, would love if this would be something examined in more detail in clinical trials. Did you want to add anything here, Chris? Did I forget anything?
No, yeah, I mean, just the fact that the normal number in patients of NKs typically of the PBMCs is anywhere from, 0 obviously to 10%. So you're fairly, the long-standing are fairly low on the normal scale, as you can see. So
yeah. And then
interesting, very interesting. And I love how you did this sort of layered approach to this, set of data.
Yeah. So the next thing, of course, when we think about genetic risk for T1D, of course, we have to talk about HLA expression. So we know HLA is, they're differentially expressed. We have different haplotypes, but here we just use flow cytometry and just use kind of a pan HLA-DR antibody. So it's not looking at DQ8 versus DQ2. It's kind of just looking at overall between healthy controls and our long-standing patient samples that we have. And this trend was pretty standard, what we saw with the small number of patients that we looked at, that when you look at the 56 brights, you can see that they tend to have more MHC, the HLA, but it's low expressing to what you typically find when you look at the live CD45 and you see that bright peak, which is probably B cells, macrophages, more traditional antigen presenting cells, where we didn't see much of this expression in healthy controls on the CD56 dim or the NK effector cells, the killer cells. And interestingly, we saw this trend in the long-standing patients where the 56 brights actually have a higher proportion of cells that are expressing HLA-DR molecules. So we found that super interesting. People don't normally think of NK cells as expressing HLA markers. And so since we know this is a risk allele for patients, it would be interesting again to look at these different subsets and look at what the HLA expression patterns are in control subjects and in T1D patients.
Yeah, that is fascinating. Are there any other disease states where this is also seen, the HLA expression in NK cells, like MS or any of the cousin diseases?
Right, I've looked, so colitis has a lot of shared risk alleles and things, but I haven't come across any papers where people have really been looking at HLA expression. And Chris and I actually kind of stumbled on this. We added it to the panel for another reason. And it was kind of like, whoa, look at how much HLA expression is on the 56 brights.
Yeah.
And if you use the c-KIT as the marker instead of bright versus dim on the 56, it's even a little bit more cleaner, the data.
Wow.
I would love it if people would start also including c-KIT when they're looking at human peripheral blood lymphocytes and looking at NK cells.
Yeah, I mean, I think this speaks to the fact that we can't find other papers. It speaks to the fact that the NKs are really understudied.
I agree. Yeah.
Because people found the cell that was infiltrating the islets, right, or the beta cells, which is the CD8s, and destroying it. But nothing happens unless these cells start to cascade, the NKs, right, and other antigen presenting cells. I mean, they're not, but like macrophages and things like that. So you have to have a triggering event.
Right.
People start looking at it. So we hope they start looking at it.
Yeah, no, I think it's a real call to action.
Yeah, that's why we wanted to show this data. This isn't actually published, but we actually would love if people would, start including and thinking about NK cells. So you can go to the next slide.
There you go.
Kind of get in the mouse model. So my lab studies the NOD mouse model for spontaneous autoimmune diabetes. And so, as I mentioned, my lab is mostly a T regulatory cell and using Treg cells as a therapy for T1D and for inducing tolerance in transplantation settings. So there was a couple papers that came out from Diane Mathis' paper and also Rodunsky's lab. Oh, in like 2014, 15, I think these papers were. And so it really came out like this is my own panel kind of piecing their data together, really showing that there's this interplay of the innate and adaptive immunity. And in panel A, when you have kind of just normal Tregs and you have an activated CD4 cell, it starts making IL-2, and Tregs with their high expression of CD25, they're really good at utilizing the IL-2 that's in the microenvironment. And then in turn, you get Treg cells to be suppressive and kind of turning off activated T cells. And you have NK cells there, and we know NK cells, especially the 56 brights, express the CD25. They express the trimeric IL-2 receptor, the high affinity IL-2 receptor expression. And Diane Mathis and Rudinsky also showed that this population expresses CD127, the IL-2 receptor alpha chain. But was also noted that when they depleted Tregs using either the diphtheria toxin model or other ways of depleting the Treg cells, they kind of got this unleashing of NK cells. And actually Diane Mathis's paper showed that you have a lot of the NK cells in these diabetic lesions when you deplete the Treg cells. And so it really was centered around the bioavailability of IL-2. So since the Treg cells are gone, there's all this IL-2 in the microenvironment, and these NK cells start using it because the Treg cells who have this competitive advantage with high expression of CD25 are utilizing most of the IL-2. So now you get this unleashing of NK cells, and they showed that you get a lot of interferon gamma coming from these cells types. So we think it's really important to be studying this interplay between the CD4, the CD8s, the Tregs, and the NK cells. Next slide, Chris. And so one of the things I noted, so my paper came out in 2020 in diabetes, where we were using anti-CD3 in combination with antigen-specific Treg cells. And one of this isn't published data, but one of the observations that also played into why we wanted to start paying more closer attention to NK cells was, if you look at the left panel, so you have untreated NOD mice. These are late pre-diabetic, so they're 16 to 20 weeks of age. So they don't have overt diabetes, but they're late pre-diabetic based on age. And so if we stimulate with PMA and inomycin for 4 hours, we see what the total interferon gamma is. And we use flow cytometry and looked at the CD45 positive cells that were penetrating into the islets. You can see that, not too surprising that you have CD4, CD8 cells, the white and the black pie, but the green is the NK cells. So you have NK cells contributing to interferon gamma production and TNF alpha just in the normal state, just in early, these mice are likely to be turning to become diabetic. But we use the anti-CD3 in the right panel. Again, we stimulate with PMA plus inomycin 4 hours. It's not too much of a guess that we have more NK cells making more of the interferon gamma because we depleted some of the T cell population with the anti-CD3. But what struck us in this study is if we just took the cells from the pancreas from the anti-CD3 treated mice and didn't stimulate them ex vivo, we saw a lot of interferon gamma production, 32.4%, and TNF alpha, 16.3%. And the majority of this cytokines, these pro-inflammatory cytokines, are actually coming from NK cells within the pancreatic islets themselves.
Wow.
And this idea of, the anti-CD3 affects you get early Treg depletion, they rebound quite quickly, but when you give the NK cells this footing to start using the bioavailable IL-2, it seems like you get this unleashing of pro-inflammatory NK cells. And so we really got even more interested in looking at the NK cells, especially using the NOD mouse model.
So Allison, just to comment on that, I know that they've tried to treat like patients with chronic fatigue and other things with high dose IL-2. That could potentially speak to this, that most of them got cytokine storm and some of the patients passed away because I guess if you're increasing that availability, even if there are Tregs present, maybe you would just start secreting the other inflammatory cytokines out of control, like you sort of show here ex vivo. Is that likely?
I mean, it could be, anti-CD3 is known to cause cytokine production. But the one that they use in the patients is the Fab, so it doesn't have the FC portion. So a lot of that is mitigated. But here we're looking at the NK cells. They don't have the CD3 on the surface. In fact, these are gated on CD3 negative cells to make sure we weren't looking at NKT cells, for example. And these NK cells are just making copious amounts of these inflammatory cytokines with T cell depletion.
Due to the IL-2 though, right?
Curious, yeah, it would
Now
be curious
available
in
to
the
them.
it would be curious in the patients that from the trial, if they have frozen samples,
Mm,
we could see a similar trend.
yeah.
But nobody looks at the NK cells in that anti-CD3 trial, so we don't know what's going on with that population. But next slide, Chris. I'm gonna skip this one, Chris, because
Okay.
we're running out of time here. Go to the one, yeah. So one of the things is I haven't published this data yet. It's getting pretty close. One of the kits we need has been on back order since the beginning of summer. Hopefully it'll get released and we can get the paper out there. But we kind of looked at these kind of idea of identifying two subsets similar to what we see in humans and seeing if we can find similar signatures. So if you look to the left, it's comparing subset 1 and subset 2. Sorry, I can't tell you what the subsets yet are, but hopefully by the end of the year, the paper will be out and I can fully tell you what the subsets are.
It's a good teaser for people to be looking for that paper.
Yes, so subset 1 looks a lot like the 56 brights. So look at all the class 2 markers, right? You have a whole bunch of where they're much more expressed in subset 1 versus subset 2. You also have CCR7, you have IL-2 receptor alpha, TNF alpha. So a lot of things that we are associating with the 56 brights for the human counterpart. And on the right, you can see the genes that are more expressed in subset 2, which you have killing molecules, you have granzyme B, perforin, again, molecules that are associated with the 56, the killing molecules.
Yeah.
So we find these two subsets in the mice very similar to what we see in humans. And you can see the green are the highlighted ones that are associated from the GWAS studies. So you have 8 or 10 GWAS genes out of 30 genes in subset 1 that are expressed and associated with T1D risk compared to subset 2, which only had 1, the green highlighted, the IL-18 wrap out of 35 genes. So subset 1 we need to pay attention to, and how is it contributing since it's associated with the GWAS studies and having a higher risk for T1D. So I just wanted to point that out before Chris wraps his stuff up with the folic acid, which I think is the next slide, Chris, right?
No,
Oh no, this is just showing you that
that same thing.
56 brights versus 56 dims.
Okay.
Do
you want me to keep going or?
Yeah, keep going.
Okay.
There you go.
All righty. So yeah, this leads into another part of our investigation. So we know, looking at literature over the last 30 to 40 years, how rapidly all autoimmune conditions have increased, particularly even type 1 diabetes, and much faster than epidemiological predictions, right? So we were very curious, like what could be causing this potential defect in the immune system, again, linking it to NKs and other things. I mean, there's a lot of hypotheses out there about, endocrine disruptors in plastics, environmental things like mold, all these different things that could do it. And you can almost find a correlation with anything, even things like, dancing to the Macarena at a wedding, or playing too many video games. I mean, all these different things. But there was one thing that struck me in particular, and another thing that I had been talking to my doctor about, which was folic acid and the folic acid cycle in general, which is critical in multiple functions in all cells in our body. I mean, the production of energy, the production of proteins and other structures, amino acids, and control of the immune system in the different molecules, the different purines that it forms at the end of its cycle, these things, inosine and adenosine and things like that. So I started looking at folic acid. I mean, I know what it is used for and why we started supplementing all wheat products with it and all enriched flours. And the idea was to reduce neural tube defects in babies, things like spina bifida and other complications. One thing that struck me is when I was walking around campus one day, I met this woman that had a tie to the DRI, and she was a nurse on campus. And you know, she had seen that I was researching some of this stuff, and she told me the story about her child that had been born with spina bifida, despite the fact that she had taken in copious amounts of folic acid during her pregnancy, much to what her doctor had asked, and even more than. And the doctor actually got upset with her when the baby was born with spina bifida. The interesting thing to me was less than 4 months later, the baby developed type 1 diabetes. So, you know, now with things like 23andMe, and there's all this research into things like the folate cycle, you can even get a report on it. And there are defects in the folate cycle that basically show improper metabolism. And in fact, it reaches out to as many as 50% of the world population. I mean, particularly people of European descent and things like that. So what we found is doing literature searches, we wrote this review and came up with another hypothesis where I was looking for anything regarding folic acid. There wasn't a lot of papers out there, but there was one that struck me in particular where they had cultured liver cells with folic acid and had seen that when they give the synthetic folic acid, they reduce the activity of the enzymes in this important pathway, particularly the very first enzyme, which is this one DHFR, where everything else cascades down from. The folic acid will reduce the activity of that 5,000-fold in all the cells that they cultured. And if a person has one of the defects in the folic acid cycle, it can be up as high as 35,000-fold. So if you think of producing energy in your cell and you're trying to make this energy coin ATP, and you're slowing down the production of all these enzymes down here, we feel that what you're basically doing is, since the cells are craving energy and needing energy, you're driving this shift towards the easier of the two pathways, which is glycolysis. And now they're learning that glycolysis and oxidative phosphorylation, the two splits, one that produces more ATP and one that produces less, are very important in immune function and drive specific functions in immune function.
And Chris, I mean, this is
Yeah.
really interesting. I mean, isn't it also true? I've talked to Amra Altidis at BC quite a bit. You know, he's really digs into the microbiome, and the microbiome is very important for intestinal folate production, right?
Yep.
So
Absolutely.
So, you know, some of the triggers, you know, have been sort of mixed reviews, whether or not antibiotics are involved or whatever, and maybe in some people they are or not. But that any disruption in that, you know, intestinal microbiome production of folic acid could also play into this type of model. Is that right?
Yeah, I absolutely agree with that. I mean, in fact, that one of the people I reached out to first, who is a person I consider a dear friend, is Mark Horowitz out of UBC.
Oh yeah.
He was looking at some intestinal cells that are very similar, like they were called mate cells, and it was actually somebody else that was looking at them originally, and then he had reached out to them, and he had a high interest in them too, and they're very important in the production of folate in the intestine.
Yeah.
So
we're featuring him soon.
Yeah, he's awesome. I mean, just a great guy and such a brilliant immunologist. I mean, he was also involved in the viral stuff as well, looking at the Coxsackie-V4 and the IF1H1 and things like that. So, I mean, essentially what we started doing was just looking at what happens when you introduce folic acid versus the naturally occurring folate. So now you can see, like in some obstetrician's office, they're offering women the choice. They'll do a 23andMe, they'll look at their folate genes, and then they'll say, well, we think you should be taking not the synthetic folic acid, but they offer methyl tetrahydrofolate, which is basically the naturally occurring. The reason why they invented folic acid and supplemented wheat products with that is that it's much more stable than the regular folate, right? So it was just a way to spray it on your cereal and make sure it was still there 6 months off the shelf, no other reason. So basically, there was not a lot of research done into what the potential long-term effects were. So now you see these papers coming out here, like the levels of unmetabolized folic acid in patients from the NHANES study, which is like the National Nutrition Study that goes on year after year. Now, granted, early on, they didn't look at all the different byproducts of folic acid. So we've gone back trying to mine some of that data, and there's a lot of pieces missing because only recently did they start looking at some of the more breakdown products of the folic acid. But without a doubt, the amount of unmetabolized folic acid in people's urine samples and blood samples has gone up 5-10 fold in the last 20 years, 30 years since the fortification program began. And it was thought that that's a water soluble vitamin. It should be, disposed of through your urine, but it's still remaining in the blood. And those levels they're finding are actually toxic. They're associated with decreased NK cell activity, believe it or not. And I think that's because they're producing adenosine at the end instead of inosine like they should. Inosine is a purine that drives NK function in the effector pathway. Adenosine, as we know, is sort of immunosuppressive. When you take things like methotrexate, right, it increases adenosine production and suppresses you. So that basically taking folic acid in some ways in the quantities that we take, if you're eating processed foods, is likened to taking adenosine. And the other thing that was interesting to me too is hearing so many people that are developing type 2 and things like that, like how they try to get off of processed foods, right? How it's so hard, how it's like an addiction. And if every cell in your body is craving for ATP because you're running it through the wrong pathway because you have that defect or you can't produce energy fast enough, it makes sense to me. It also increases improper lipid storage, right? So you can't do anything with all this glucose. So you're putting it away as instead of breaking it down into ATP.
I love how you're talking about this from a physiological standpoint and really trying to incorporate all the different pieces, not just sort of like one target approach. It's really, really fascinating.
Oh, I appreciate that. I mean, yeah, a lot of it is hypothetical, right? But I want people to think about it.
Yes.
Because I know there's people out there that trust me, I couldn't break down any part of this pathway and understand it. I'm not that much of a biochemist, but people that can start looking at it and figure it out, because we need to fix these problems that keep getting worse.
Yeah, show the next graph.
Yeah, so here was the thing that struck me, like when I was, and it's funny because I was talking to one of the physicians here and he's like, yeah, like I said, you could do that with video games or anything. I said, yeah, but video games don't directly affect the energy pathway the way folic acid does. But this is just some data showing the trend of diabetes in the country, both type 1 and type 2. But starting in, and then at the point where folic acid fortification began, you see this tremendous uptick that wasn't expected. And at the same time, we have the levels of unmetabolized folic acid that are shown in the little green bars that went from 5.8 up to 13.8 and higher. And now they're well above 20 to 25, I believe, is what people are saying. So, it's in every product. It's hard to find flour without folic acid. That was something I was trying to do, was reduce my intake of it, for example. And even now they're putting it in things like rice and salad dressings and things where you wouldn't expect to see it. And I can't imagine that. And in this country, we really didn't have a problem with deficiencies in folic acid. That's more in developing countries, right? But now even in developing countries where they didn't see incidences of type 1 diabetes, that's all increasing too in other places.
This is really fascinating, and I'm going to share it with a couple people directly, just because scientists, because I think I've been talking about them sort of around and around different parts of this, and I think it'll be really interesting to them. One thing I did want to just totally share with you guys anecdotally is, I formed this group called the Sugar Mamas. They had like 40, moms in there that had kids with type 1. One thing we talked a lot about, again, anecdotal, that a lot of the kids, almost the majority, had recalcitrant warts, which is associated with natural killer cell deficiency. It's so interesting, right? That was also just kind of something that was just talked about. And perhaps, right, that was sort of like lurking in the background. Who knows, what prompted this NK deficiency, but one pathway certainly could be this dietary pathway.
Yeah, and warts are often of a viral origin too, right? That's another thing that we forget, so it makes sense. Yeah, who knows how it starts? I hear a lot of parents anecdotally talk about how their kid had a very severe infection of one sort or another, right? So whether it be a bacterial, viral, like a very difficult infection. And if you think in terms of when I got the vaccine, that wasn't what prompted me to get diabetes, but it's the same sort of thing, right? Like if you get an infection, your immune system is concentrated on that. If you get a vaccine, your immune system is concentrated on that. It gives an opportunity for all this stuff to pop up. So now I feel like I'm playing a chronic game of whack-a-mole, trying to knock one down, the other one pops up, right? So once you get that balance out of whack, these things can happen. So that's the sad thing to me. So here's a little bit of data that we produced in, just studying this stuff. We took just the synthetic folic acid, and we actually used that in an insulin stimulated glucose uptake of muscle cells, which are the main users of glucose. And we used 2-deoxyglucose, which allows you to study it because it's well labeled and you can measure measure an output. And so essentially what we saw is that when given insulin, the higher the dose of folic acid, the lower the difference between stimulated and basal uptake. So essentially, like when you have insulin on board, they're just not consuming the glucose because there's some sort of disruption, and it's probably because it's just shuttling towards glycolysis and they're not taken in. So we see that your insulin sensitivity is essentially suppressed by this. At the same time, we did another thing with the insulin tolerance test and looked at the blood glucose drop in animals that were fed just to control folate versus the folic acid. And we see again that in the folic acid, there was a trend for the glucose to go down less per minute with the same dose of insulin than in the control group. So they're not using it efficiently. It's not consuming the glucose. And these were on a high dose of folic. I think it was the 40 micromolar dose.
So the folic acid really could be contributing, a big contributor for those with type 2.
As well. Yeah, I mean, I think it's a metabolic
yeah.
cycle that affects everybody, right? Like, so you can't, every cell utilizes that cycle to produce energy, to control immune response in some ways, as we know now, Allison, from the stuff you guys have been looking at with different metabolic effects on immune function and cytokine production and things like that, whether it be pro-inflammatory or sort of regulatory. Yeah, I think it's a critical cycle, and that's why this one struck me more than like playing video games or other things like that. So in my lab, I just like to thank people that have been instrumental during the time, besides Allison and everybody in her group, but like LeBaron, Agostini, one of my graduate students, Graham Gardner, a postdoc, Paula, who was an undergrad, Jessica, who also worked in Allison's lab as well as an undergrad, and then one of my other postdocs, Mohammed Hossein Tutuji, and then finally my colleague Armando Mendez, who is the biochemist that helped me with a lot of the glucose uptake studies and insulin tolerance tests and things like that. He's an endocrinologist, biochemist. And then Allison, I defer to you.
Well, I mean, it was really Chris, like, not coming down from the 5th floor to the 2nd floor in the DRI. And it's like, I have this crazy idea. What do you think? And it's, just through conversations with Chris, I'm like, Chris has a really good, he undersells himself. Chris has the great ability to think outside the box and put the puzzle together. So if you ever have an opportunity or think you have a crazy idea, contact Chris Fraker and bounce that idea, because he's very good at putting the pieces together. So yeah, and really this paper that I've been working on for the last 2+ years now really stemmed from initial conversations with Chris and trying to understand what NK cells, and of course I'm interested how they impact Tregs and how they may negatively impact Treg based therapies if we get these inflammatory NK cells, because we know natural Tregs, don't like those environments. And yeah, so I would
I would also just offer that I just love the fact that the DRI Miami really fosters this kind of out of the box thinking and this collaborative environment, and is very supportive of really trying to push the knowledge base forward through both collaboration and innovative ideas. It's so important, and it's kind of rare these days to get that kind of an environment. A lot of times people are so preoccupied on the next grant and whatever that they kind of stay in their lane very, very much. So I think this is a very organic and exciting environment at the DRI.
Yeah, I mean, and speaking of that, we did try to get funding, through external sources, but I mean, at least I tried a couple of times, and Allison is now getting some because she's her work obviously isn't much. I did it more on some of those innovative things like for young investigators, and it was hard, but the DRI Foundation always was able to keep us going and give support to this because they, you know, they saw promise and interest in it. And now I think Allison, you know, she'll have funding to do some of that work looking at NKs in the future too. And so I'm excited about it. And, you know, we've talked to colleagues at places like UF too, and tried to get some of them excited. And I have a colleague at University of Alabama, Birmingham, and Mark in British Columbia. So, you know, as I keep blabbing through Zoom or whatever, I'm just pushing, because for me it's personal, right? I'm like, come on, start looking at that. And that's what I did to Allison too. So when she says I came down with a crazy idea, she was like, Chris was crazy and came down and bugged me and wanted me to start working on it.
Yeah,
Because
well,
I want it cured, you know, that's the motivation.
yeah, well, we know what people like Steve Jobs says about crazy ideas, that they often lead to breakthroughs. So I think that
I hope so.
I think there's nothing crazy about these types of ideas that you're talking about. They're really innovative and they're very interesting.
Yeah, I think in clinical trials, people just kind of think of NK cells. Like I said, people just think of the killers, the NK effector cells, but it's really the NK regs, the 56 brights that are making all the different cytokines. They can actually make inhibitory cytokines, IL-2. You know, so they play a big role in the adaptive response and what kind of response you get.
Yeah.
Just kind of blanketly look at this as like a big, like one big population, but it's actually very heterogeneous. And we know NK cells belong to the innate lymphoid cell group 1, right? So you also have the traditional ILC1s that are often ignored and make a lot of interferon gamma. And we look at that subset too, or see some really interesting things. So, you know, I'm at the point where I've had some funding for it. And, you know, like I said, a lot of our foundation has continued to when we haven't been able to get funding, and because they believe in our ideas and, you know, we feel passionate about it and we need to get the word out. So like I said, I'm a Treg person, but, you know, got into innate cells and NK cells.
Well, it's great to have this cross collaboration. I think in the interest of time, we may stop here, but I'd ask you, anyone in the audience that has a question, to reach out directly to either Dr. Allison Bayer or Dr. Chris Fraker, and ask them via email, just because I'm sure they have a busy day to get back to. And we did go over quite a bit, but it was so fascinating, and I so appreciate your time.